Hi everyone, and welcome to our review on carbohydrates. This is actually going to be the first in a little series of videos about carbohydrates, because what I've done is I've split it down into basically the different categories, if you like. So we're going to have a look at the monosaccharides in today's video. As far as the specification goes, what you need to be able to do is describe that ring structure of glucose and its properties. And this is as an example of a hexose monosaccharide. You also need to recall the structure of ribose as one of those examples of a pentose monosaccharide. A little bit more detail that's in that little to include bit on the specification, and that is that we need to know the structural difference between alpha and beta glucose and the difference between hexose and a pentose monosaccharide. Before we delve into the actual structural side of things, it's worth having that little moment to recap on what we actually use carbohydrates for. And there's basically three categories of use of our carbohydrates. The first one is that we use them as a source of energy, and best example that you would have heard about so far is good old glucose. Second one is that we can use them as a store of energy, and back in GCSE you would have definitely heard about starch and glycogen there. And finally, we use them as a structural unit. So this is in things like cellulose in our plant cell walls, or chitin. So three uses of our carbohydrates: energy source, energy store, and structural units. First thing we really need to consider here then is to understand what on earth we mean when we use this phrase monosaccharide. So if we actually just break that word down into its two parts, so you literally just split it there, mono just means one, and then saccharide refers to sugars. So what we see with the word monosaccharide is we're talking about one sugar, so a single unit of a sugar. That's all the word means. So what we're really talking about as far as the monosaccharides go, it's the simplest form of a carbohydrate. They are single units, therefore they won't be any simpler as a carbohydrate. Now, what you may realise is that you've actually heard about a range of these carbohydrates previously. You just might not have realised that that's what we were talking about. The way you can identify some of these is because their names end in OSE. So think about some of these you may have heard of: lactose, maltose, sucrose. Those are common carbohydrates that end in that OSE that you probably have heard about previously. In terms of that major use that we mentioned earlier, this important source of energy, what we find is these monosaccharides are going to be one of the things we spend quite a lot of time focusing on when we come to the respiration part. And what we will do is have a look in detail at how we actually take our glucose molecule and then we convert it into quite significant amounts of ATP. The last thing to look at on this slide is just how we can work out the number of carbon atoms that are present. Now, this is just going back to one of those basic principles of naming. So what we do know is, I would hope, that there are two that we have to look at on here. We have hexose and we have pentose. So those are the two we mentioned in the specification at the beginning. Now, I'm hoping that from a bit of maths knowledge that we know the word hex means 6. So any hexose will have 6 carbons in it, whereas pent means 5. So pentoses have 5 carbons in it. We could come down and have a look at something like a triose, for example. Tri means 3 carbons. So you can see how looking at the actual first part of the name gives us the number of carbon atoms present. So the two to really remember there are obviously hexose, 6 carbons, pentose, 5 carbons. And we'll look at those in more detail as we carry on with this summary tonight. When it comes to the structure of our monosaccharides then, we've got two options really. We can have straight chains or we can have ring forms. So when I'm talking about a straight chain, what we find is that the carbons all align in a straight chain, as the name suggests, and then we're going to have our little hydrogens and hydroxides attached all the way down, etc. Whereas the ring form is where we're actually going to end up in this little structure, and I'm going to draw just a basic one at this stage, with the carbons all arranged in a little ring. We will have a little bit off here, I'll put that one in. So we've got those two different varieties. You either have the straight chain form or the ring forms. When we've got a molecule that has the same formula, but the atoms are actually arranged in a slightly different configuration, then we have something we refer to as an isomer. And these isomers are going to be present whether we're talking about straight chain forms or ring forms. These two diagrams here just show you the two straight chain forms of glucose. So we've got L-glucose on the left, D-glucose on the right. And if you look, actually, the vast majority of the structure is the same. They've both got the same formula overall, but when we look at it, if we start at the top of the molecules and just work your way down, you can see that a large proportion of this is identical. So we've got the carbon double bonded to the oxygen and a single hydrogen at the top. Then we're going to come down our carbon backbone chain, and then we get to the bottom, we've got CH2OH off the other end. The difference comes with the position of the hydrogens and the hydroxyl groups. So if you look at in this case, we can see that we have a hydrogen on the left, whereas a hydroxyl on the left of the L-glucose. And then if you carry on down, you can see that we get that little repeating pattern where we've got those two groups the other way round to one another. If we look at alpha glucose in a touch more detail, it's glucose, therefore its formula is C6H12O6. And the key roles for our alpha glucose is it's an energy source, first of all, but it's also going to be a significant component when we come to look at the structures of our starch and our glycogen. So there's its formula again, just as a reminder for us. Beta glucose, same formula, still glucose, C6H12O6, still an energy source, but this time the beta glucose is a component of cellulose. Make sure you learn which glucose isomer matches which polysaccharide, because again, this could be a nice easy question just from a recall perspective. The specification did tell us we need to know about our little pentose sugars as well. Now, pent means 5, so we are looking at 5 carbon sugars. And we'll have a look at 2 examples of this. First one is ribose, so formula because it's got 5 carbons, C5H10O5. Now, ribose you could well have heard about because this is a component in RNA, in ATP and in NAD. So 3 very important molecules in living things, and ribose is found in all of them. You could be asked to draw the structure of the ribose, and as far as I can recall, the only question that has asked this has been focused on where the carbons are, not the hydroxyl and the hydrogen groups. So the rules for drawing are pentose. We start with the oxygen at the top. We're then going to draw our ring, which has got 1, 2, 3, 4 carbons in it. And then what we do from the carbon to the left of the oxygen is we're going to add our CH2OH group. So the rest of it, we just be filling in the hydroxyl and the hydrogens. But if you're just asked to lay out where the carbons are, that's all you'd need to do, because we've got the 4 in the ring structure with the oxygen, and then your 5th carbon coming off from obviously that 4th carbon just before the oxygen there. Final thing is our deoxyribose then. Now we can work out a little bit about this by the first part of the name, deoxy. So if we've got deoxy, we've removed an oxygen. So it's still a ribose, so still 5 carbons, still 10 hydrogens, but rather than having 5 oxygens, because it is deoxy, we've removed one of them, it's just O4. And deoxyribose is one of those components of DNA. So when we actually look at it, you'll see that the structure is identical to the ribose, with the exception of when we have a little look over here on our 2nd carbon, we've got 2 hydrogens rather than a hydroxyl and a hydrogen. So that's the only difference. We've removed one of the oxygens, therefore deoxyribose. As always, if there are some bits that you'd like to go over in a bit more detail, then there are some extra resources over on the website. Link is in the description box underneath. And of course, don't forget to subscribe so you don't miss any of the future updates.